Hello everyone, welcome to NEET YUG. I am Arun Kumar. Here I am going to explain cyclic photophosphorylation. It is the process of synthesis of ATP within the stroma lamella with the involvement of 700 nanometer of red sunlight and photosystem 1. That's it. Here the photosystem 2 is absent and the enzyme, that is ferredoxin NADP reductase, is also absent. Hence, it cannot synthesize or produce NADPH, only ATP synthesized. Phosphorylation means it is the synthesis of ATP from ADP and inorganic phosphate. Photophosphorylation means with the involvement of sunlight, that is 700 nanometer. Okay, so this is the model of one thylakoid. That means I will be talking about non-cyclic photophosphorylation for now. You could see two systems here that is embedded within the membrane of the thylakoid. This is PS2, this is PS1. Okay, here the water will be there, that is the lumen of the thylakoid, and what's going to happen is a splitting up of the water takes place and electrons will be accepted by this PS2 and it will be transferred to ETS, and at the time ATP synthesized, then it is taken to the PS1, then the PS1 actually transfer that electron to the FES, that is iron sulfur containing protein, then ferredoxin, then FNR, and finally NADPH is synthesized, that is in the stroma. Got it? So these are actually the things about non-cyclic photophosphorylation, but in this case PS2 is absent, that is cyclic photophosphorylation, and the site of cyclic photophosphorylation is entirely different, not in the thylakoid, it is in the stroma lamella. Okay, so let us see. We just draw the thylakoids. These are all the thylakoids. Make it grana, and you know that there is the connection, the physical connection between the grana is called as the stroma lamella. Correct? So this is now the stroma lamella. Now in the stroma lamella, what is there? The PS1 is there. That means P700. Photosystem 700 is there. That means it uses 700 nanometer of wave, that is sunlight, wavelength of sunlight, that is red light. Okay. So 700 nanometer, that is red. And another thing is there is no FNR. The enzyme NADP reductase is absent. And one more thing is there is no PS2. The system PS2 is absent. Got it? Compared to non-cyclic photophosphorylation, cyclic photophosphorylation, the rate of photosynthesis will be lesser. Got it? Compared to non-cyclic photophosphorylation. Why the rate of photosynthesis is less? Because beyond 700 nanometer of sunlight, the rate of photosynthesis will be less. It was observed by Emerson. Hence, it is called as a red drop or Emerson's effect. We know about it. Okay, now I am going to give you the detailed structure of cyclic photophosphorylation. For that, I am going to draw one circle and let me just name it as light harvesting complex, that is nothing but PS1 or P700. Okay, so the splitting up of the water occurs now in the stroma and which is accepted by this PS1. Yes. Okay, then it gets excited. What get excited? The chlorophyll A get excited. Now it transfers the electrons to the ETS. Same, ETS means what is there? If you see the ETS, here we have the phyophytin, then comes the plastoquinone, then cytochrome B6F, then plastocyanine will be there, and the electron finally reaches LHS. That means you see only the circular movement of the electrons within this photosystem 1. Hence it is called as cyclic photophosphorylation. When the electrons are moved into the ETS, what's going to produce? ATP synthesized, that is phosphorylation. Got it? Okay, so using ADP and inorganic phosphate, ATP synthesized here in the ETS. This production of ATP can be explained in chemiosmosis. Okay, that we'll discuss it later. So this is all about cyclic photophosphorylation. Hope you understood this session. In our next session, I'm going to discuss about chemiosmosis, how exactly ATP synthesized within the electron transport system. If you like the video session, please subscribe to our channel. Thank you.